Okay guys, so this is going to be Chapter 6, Part 1, Cells Overview, Nucleus, Ribosomes, Mitochondria, and Chloroplast. So let's go ahead and start filling in that middle part on top of your notes. Let's answer the question, what are similarities and differences between prokaryotic and eukaryotic cells? Well, all cells have a plasma membrane, cytoplasm, DNA, and ribosomes. Both prokaryotic and eukaryotic cells sometimes have cell walls, cilia, and flagella. Prokaryotic cells have a DNA in region called a nucleoid, ribosomes, no nucleus, and no membrane-bound organelles. And bacteria and archaebacteria are both prokaryotic. Go ahead and label this on your cell. It definitely needs to be labeled by tomorrow in order to do our fun little sugar experiment. So press pause if you need to. I'm moving on. Eukaryotic cells have a nucleus containing DNA, membrane-bound organelles, and they're usually much larger than prokaryotic cells. Examples are like protists, fungi, animals, and plants, which all consist of eukaryotic cells. So what limits cell size? Like, why are they so tiny? Well, surface area to volume ratios limit that size. Larger cells have very small surface area to volume ratios. So this means the larger cells are less efficient at transporting nutrients, oxygen, CO2, and waste across their membrane. So why are eukaryotic cells able to grow about 10 times larger than prokaryotic cells without starving? Okay, well, let's go ahead and see what they do. So eukaryotic cells have membrane-bound organelles that increase their internal surface area, including mitochondria that make ATP, chloroplasts that make glucose, and the endomembrane system, which is Golgi, smooth, rough ER, vacuoles, lysosomes, and vesicles. These are going to allow eukaryotic cells to package, transport, digest, and store substances more quickly. So for example, if I look at this mitochondria, you know how it always has folds? Well, the reason why it has folds is to increase the internal surface area. When we talked in 7th grade, we said this looked like pancakes, but actually it's just folds that are happening back and forth to increase the surface area. Golgi apparatus, the rough ER and the smooth ER, they all have folds. Why? To increase the surface area. So let's talk about microscopes. How do we use microscopes to study cell structure? Well, light microscopes can be used to magnify an image about 1,000 times, whereas electron microscopes shoot little beams of electrons, which basically kill the specimen. You have scanning, which shows the surface, and then you have transmission, which shows the inside of a specimen. So here's examples of a light microscope. Look at how big this eukaryotic is compared to this prokaryotic. Do you see these little blue squiggles? These are all like prokaryotic. This is huge. This is tiny. This is huge. This is tiny. Okay, let's keep going. Here's some more scanning electron microscopes, scale worm, pollen, so cool. Here's like intestines, here's our body fighting some bacteria, awesome. Then we have transmission. Transmission is showing the inside, and look, we have more folds. Why do mitochondria have folds again? To increase the surface area. And over here we have like an immune cell right here, and it's eating this yeast. So let's look at these pictures. Can you tell which one is scanning, which is looking at the surface, and which one is transmission, which is like looking at the internal structure? I'll give you a second. Seconds up. This one is scanning because it's the surface. This is transmission because it's on the inside. So let's talk about cell fractation. This is when you want to study a specific organelle, maybe just to look at it, maybe for a disease. I don't know. It's up to you. So in this one, we homogenize the cells by putting them in a blender to break them up. Then we spin this at high speeds in a centrifuge. The cell organelles separate based on size and mass, so the larger and heavier organelles will sink to the bottom. The process allows us to separate and observe different organelles. So if I'm looking at this picture, can you guess which ones are heavy and which ones are light? So check this out. I know this is kind of hard to see, but you can see like cytosol, for example, is on the top. That's because it's what? Is it heavier or lighter than ribosome? It's going to be lighter because it's floating towards the top, and ribosomes are heavy, so they'll be towards the bottom. So anything on the bottom is going to be the heavier organelle. All right, let's go ahead and keep going. What's the structure and function of the nucleus? Well, the nucleolus is the dark area where ribosomal RNA is built and produced, Ribosomal RNA is produced and ribosomes are built. So the nuclear envelope is going to surround the nucleus, and then you have these purple little like holes that are nuclear pores. that allow RNA and ribosomes to exit the nucleus. And some are directly connected to the rough ER, just as you can see like right here. So go ahead and like label this. If you need to pause it, go ahead, but I gotta move on. Make sure it's labeled for tomorrow. How is DNA stored in eukaryotes? DNA is first wrapped around little proteins called histones. They look like beads on a string. Then these histones get together and they get organized and they make chromatin. During mitosis, chromatin condenses into X-shaped chromosomes. Each chromosome consists of two identical sister chromatids connected in the middle by a centromere. We can't see it here, but the centromere would actually be right here. So go ahead and take a second, pause if you need to. Label this diagram on your notes because I gotta move on. So how is DNA stored in prokaryotes? Well, DNA is actually naked. It doesn't have any proteins. It's found in the nucleoid region of the cell, which is like usually a central region, and it forms one large circular chromosome instead of like these X-shaped like strands. And prokaryotic cells can also contain smaller circular pieces of DNA, which are called plasmids, that they can actually get from other bacteria. So let's look at the structure and function of ribosomes now. Now we're looking at a very small organelle. Ribosomes create proteins from mRNA. They're found in all cells, and they're even found inside Mitochondria and Chloroplasts because at one point, those were their own cells. They have a small subunit and a large subunit. Free ribosomes float around in the cytosol, whereas bound ribosomes are embedded in the membrane, such as like on the rough ER. So why is DNA coding for a small subunit rRNA used to code or used to construct phylogenetic trees for organisms that diverged billions of years ago? We're actually going to go over this, and it has to do with the endosymbiotic theory and evidence that supports it. So mitochondria and chloroplasts probably descended from prokaryotic cells that were engulfed by a larger predatory cell. Over time, they evolved to survive inside the predatory cells and formed a symbiotic relationship with them. So here's evidence of mitochondria and chloroplasts. They have a double membrane. They contain ribosomes and circular DNA, so they have their own DNA, and they actually reproduce by binary fission, which is exactly like bacteria. So the question is, why is a double membrane a piece of evidence that supports this theory? If you can't think of something, we're actually going to get to see this with an Easter egg. It's really neat tomorrow. Make sure you have this written down. So what evidence can be observed in this image? Well, check this out. It has its own mitochondrial DNA, and it has its own like nuclear DNA. That's weird, right? Something else's DNA is inside your body. Whoa. Okay, let's go ahead and keep going. Let's talk about the structure and function of the mitochondria. Mitochondria break down glucose and other organic molecules to produce ATP during cellular respiration. They have an inner and an outer membrane with an intermembrane space between them. The folds in the inner membrane are called cristae, and they increase the surface area. So those folds that you saw help to increase the surface area. There's a space where we have ribosomes and we have DNA, and it's circular, just like the bacteria. So why is it so advantageous to have so many folds? It's to increase the surface area. So go ahead and label this. If you need to pause it, go ahead, but I gotta move on. Let's talk about the structure and the function of chloroplast. So chloroplasts can produce glucose and other organic molecules from CO2 during photosynthesis. They consist of an inner and outer membrane with an intermembrane space in between. A fluid called stroma is in the center with DNA and ribosomes, and coin-shaped membrane sacs called thylakoids. We call those like pancakes. Then you have stacks of thylakoids that are called grana. So go ahead, please label the chloroplast. And again, I ask you this question. Why is it advantageous to have so many folds in the thylakoids? Look, they look like folds here and there, folds here and there. Why? Because we're increasing the surface area. All right, with that being said, please make sure all your notes are filled out. We definitely have 2 labs tomorrow, and we have a little demonstration with yarn. I think it's a lot of fun, so please make sure everything is filled out and labeled. Uh-oh.